Welcome children. In the previous videos, we have studied about or we have tried or get an insight into the process of the sexual reproduction or various events that are taking place when the sexual reproduction is happening within an organism. The various steps we have discussed. Now this chapter 2, it is introducing the sexual reproduction in the case of plants. Now we all know the angiosperms are the flowering plants and the main site of the fertilization or the purpose of the flower is to support or have the fertilization. So flower is the site of fertilization where the formation of the haploid gametes take place over here, and also the fusion of these gametes happens within the flower. And we all are familiar with the very basic structure of the flower, that the flower, a complete flower has four whorls. That is, the outermost is the calyx, and individual is known as that singles is known as the sepals. Then we have the corolla or the petals, and the male, that male reproductive part, which is known as the Enrotium, and the female reproductive part, which is known as the Gynoecium. So in this video, we'll be discussing about the male reproductive part, that is the Enrotium. Enrotium actually happens to be the male reproductive part, and it consists of, if we see, the stamens. It is the third whorl and it has the stamen. The stamen is also having a filament and the anther. So when we talk of the male reproductive part, the Androecium consists of stamen. Now this stamen has two parts. The function of the stamen is to produce the pollen grains or the microspores, or these are also the male gametes. These male gametes are haploid in nature. So the microspore or the pollen grains are the male gametes which are being produced within the stamen. So the structure of the stamen, when we try and understand, it consists of a stalk or the filament and the anther. Here is the diagram showing you the stalk or the filament and the anther over here. The stalk or the filament actually helps in the attachment of the stamen to the flower. So we have divided this into distal end and the proximal end. These are the two ends of the filament, and the proximal end is either attached to one of the petals or the petal of the flower or to the thalamus over there, so that the stamen remains fixed or grounded in the flower, well placed. The anther is the site, or it has the pollen grains. It is the site where the pollen grains are produced. If you see the anther structure, we find a longitudinal groove running along the anther. Here, this is the site for the dehiscence. Dehiscence is the split opening of the anther, releasing the pollen grains when they are mature. So here we are going to study about the structure of the anther in detail. This is the structure of the anther, and the anther may be arranged variously in a flower, because different kind of flowers may have different arrangement of the anther over there. Now coming to the structure of the anther, when we discuss, an anther consists of it can be Bi-Lobed. It is, that is, if we see the anther structure, the anther is Bi-Lobed. And when we talk anther is Bi-Lobed, that means each anther this is lobe 1 and this is lobe 2. So it is a bi-lobed structure and it is diathecous. Diathecous means each of the lobe has two compartments. This is compartment 1, this is compartment 2. So each lobe has two compartments and therefore the typical angiospermic anther is said to be bilobed because it is having two lobes over here, and also it is diathecous or two chambered. These are the two chambers. This is chamber 1 and this is chamber 2. Now, this anther actually is having a longitudinal groove as explained earlier, and this groove causes or is the site where the dehiscence will take place. Now the bi-lobed nature of an anther is very clearly in the transverse section. If we study the transverse section, now if this is the anther and if we take out the transverse section of this particular anther, a mature one, we find very distinct arrangement of the two lobes over here. This is lobe 1, lobe 2, and these are the two chambers. The chambers are known as pollen sacs. That is, these structure, that is the two pollen sacs over here, has the pollen grains stored within. And also there are microsporangia present inside. That is 4 microsporangia. That is the plural, and it is located at the corner. So here are the 4 microsporangia which are present within the uh, pollen sac, and these are the actually sites where the formation of the pollen grains will be taking place. Now let us talk about the detailed structure of the microsporangium. Now when the transverse section is seen, it consists of 3 layers. The 3 layers are, first let us talk about the epidermis. This is the outermost layer. This epidermis is protective in nature. This epidermis is the outermost layer and is protective. The endothecium layer is the second layer. Endothecium is the second layer, and this endothecium is again protective in nature. It helps and protects the inside tissues and also forms the boundary of the anther, the towards outer side. The third layer is the um, middle layers. This third layer may be 2 or 3 cell thick, and this tapetum is the innermost layer that borders the microsporangium. So these are the 4 microsporangiums, and the tapetum is the innermost layer. Tapetum is typical and it has a very specific role. Tapetum actually provides nutrition to the developing pollen grains. It is actually a multinucleate or binucleate. All the other cells are diploid in nature, but here tapetum is also binucleate or multinucleate and has dense cytoplasm. The reason is the tapetum undergoes endomitosis. Endomitosis, and due to endomitosis in tapetum, this endomitosis causes division in the cells, and these cells form there is karyokinesis, but there is no cytokinesis. That means the cell does not divide, and as a result it is rich in nucleus and has dense cytoplasm. The cytoplasm of tapetum has other organic molecules, as well as it is rich in protein. This actually is important to have dense cytoplasm within the tapetum, because later on it helps in the formation of sporopollenin, which is a protein coat important for the pollen grain formation. So sporopollenin is actually a protein which is being formed by the dense cytoplasm of the tapetum, and tapetum is typical as it is binucleate or polynucleate, as due to the reason that it undergoes karyokinosis but does not undergo cytokinosis, that is the breaking or the cell division. Now when we talk of the other structures, there is stomium which is present over there within the this anther. The stomium is actually a common area of dehiscence. Dehiscence, again, as I told you, it is the site or it is the rupturing of the anther so as to release the mature pollen grains. It is made up of thin walled cells, and when the tissues dry over here, when the pollen grains are mature, the tissues dry, and as a result, it breaks open, causing the release. Now, let us also talk about the cells or the structures which are present within the tapetum or the microsporangium. Now, here these are several sporogenous tissues. Each sporogenous tissue is a diploid structure. It is also known as microspore mother cell. Now, these sporogenesis tissue are microspore mother cell or pollen mother cells. These tissues undergo the process of meiosis. They are having a ploidy of 2n, that is diploid structures. They undergo the process of meiosis, and each sporogenous cell will produce 4 haploid cells, and these 4 haploid cells are together called as microspore tetrads. These microspore tetrads are formed within, and they are joined to each other with a connective tissue. When these connective tissue dehydrate, they form the pollen grains. And these pollen grains on maturation are released by the dehiscence of the anther. So here is the structure of the microsporangium, and you can see that the structure consists of the 4 layers, that is the epidermis, the endothecium, the middle layer, and the tapetum. Tapetum is the innermost layer, whereas epidermis is the outermost layer. This microsporangium consists of the sporogenous tissues, which are diploid structures, and these diploid structures on meiosis will form the 4 haploid cell structure called as microspore tetrads. Then further dehydration of these tissues, surrounding tissues over here, will lead to the formation of the pollen grains. In the next video, we are going to study about the structure and the formation of the pollen grains in detail. Thank you.